everybody. This is our Lab 7 photosynthesis walkthrough. Just want to preface this with, this lab is pretty much like a worksheet. There's not a whole lot of hands-on activity that we're going to do. And so that's why I'm going to be doing it from my computer. And so what I wanted to show you guys is I'm going to share my screen. I wanted to share with you, let's go up here. I found this PDF editor online. If you guys haven't found this, this is extremely useful. Myself up here. It's extremely useful. So it's smallpdf.com /edit/PDF, or just search smallpdf.com. I'm sure you'll find it. And what you can do is you can just upload your PDF. I'm going to grab mine down here. I've already downloaded it as a PDF from our Google Drive. And so I'm going to just drop it in there. And then what's really cool about this is that I can edit directly the PDF. I'm going to zoom out just a little bit. I can edit the PDF directly, and I can like write on it with my freehand. I can highlight text. I can add text to it. And so for some of these labs, if you guys don't like printing them off, you can actually use this to do your labs. So if you haven't done this yet, I highly recommend watching one of the 3 videos here. Also make sure that you've watched the photosynthesis lecture video before you do this lab. Otherwise, you're going to be like, what are we talking about? So this lab, the Amoeba Sisters one is pretty good. But this lab, we're going to basically just kind of, some of it will be review from the photosynthesis lecture. But I'm just going to walk through it with you guys together. If you want to do this in the PDF editing program with me, that's great too. You can do that. So it says here, the leaf is the organ where photosynthesis takes place. And so what we're looking at here is a cross section of the leaf. You've got palisade cells, which are the cells that line the upper epidermis. They're at the very top. So these are the cells that receive the most sunlight from the sun. And so, of course, they're going to have the most chloroplasts per unit of area as any other cell in the plant, meaning that the palisade cells are going to have a high concentration of chloroplasts. What we want to do is we want to color the chloroplast cell, or color the palisade cells green. So I'm going to come up here and do my editor. And we're just going to color in all the palisade cells green. Now you guys can take a little bit longer and do this a little more neatly. You can pause the video if you need to. So we're coloring the palisade cells green. They line the upper epidermis of the leaf. And then the guard cells are down here on the bottom of the leaf. There will be some guard cells on the top of the leaf, but there's guard cells. Oh, hold on a second. All right, just a brief interruption. My kid's Kindle died. So such is life on this online forum that we're on. Anyways, so we're coloring the palisade cells green. Now the guard cells, we're going to color them in orange. So the guard cells are these little cells down here. I only see one. But the guard cell is what opens and closes and allows gases to get through the leaf into the palisade cells. So gases like carbon dioxide that the palisade cells are going to need for photosynthesis, but also the guard cells are going to open up and allow that the extra oxygen that's produced from photosynthesis is able to escape. Whenever they open those, sometimes water escapes as well, and so you got to be really careful. The plant needs to conserve water, and so maybe it'll only open its guard cells at night if it's in the desert when it's cooler. All right, so moving on, we colored those green, orange, moving down. So palisade cells have chloroplasts. And so what we're going to look at is we're going to look at the structure of the chloroplast. And the chloroplast is the primary organelle that conducts photosynthesis. Now, inside of the chloroplast, there are thylakoids. I'm going to try to capture these in the same screen if I can. Okay, in the chloroplast, there's these things called thylakoids, and they're basically surrounded by a membrane, and these are where the light reactions take place. So there's light reactions and there's dark reactions. The light reactions require light. The dark reactions require no light. They can occur during the day, they just don't require light. Inside of the thylakoids themselves, you got a good chance to look at this when we were doing our lecture video, is there's going to be photosystems embedded into the thylakoid membrane that helps to capture sunlight at certain frequencies of radiation. And so these are those light sensitive plant pigments that help to absorb the light and energy at a given frequency, and then help to use it to like excite electrons. And then those electrons, as you know, like this is our whole metabolism thing. If you can kickstart an electron and give it a bunch of energy, then you can take that electron and it's got free energy, and now you can transfer that free energy to something else. You can transfer it to a cellular battery and charge it up, or you can transfer it to, in this case, it's going to be a cellular battery. You can transfer it to an enzyme, things like that. You can transfer it through like a pump, like an actual pump inside the membrane, and use that to pump the hydrogens across the membrane and stuff like that. Okay. Hold on, buddy. Hold on, I gotta find my, hold on, buddy. Let me find my, I don't know how to pause my video. There, let me pause real quick. All right, sorry. Kids. So anyways, so what we're gonna do in this one is we're gonna color the thylakoids green, and we're gonna label the stroma blue. So the thylakoids are these stacks of discs that go to form the granum. So the granum is just a stack of thylakoids. So we're coloring the thylakoids green. You guys can spend a little bit more time on this. And then we're gonna color the stroma blue. And so the stroma is actually where the dark reactions take place. And so that's kind of this cytoplasm-like liquid that surrounds the granum. So we're going to color those in blue. And there's our chloroplast. All right. So like we said, photosynthesis is the most important chemical process on Earth. I know it's cliche, but it is, because that's really essentially where all of the free energy that powers all of the biology on this planet comes from, is photosynthesis. I mean, we do have like geothermal vents and stuff like that, but that's not what we're talking about. And that's a very small fraction of the energy on the planet. Let's see. So photosynthetic organisms can convert light energy into chemical bond energy. Let's see. All right. So in its simplest form, photosynthesis is a process that takes higher energy that creates high energy organic compounds from low energy inorganic compounds by using sunlight. So the most common form of photosynthesis takes carbon dioxide and water and turns it into carbohydrates and oxygen. That's sort of like an 8th grade level understanding of it. The basic equation is just this. And so remember that this equation is sort of a general equation. And what we could do is we could zoom in here and break each part of this equation down into steps. We're not going to do that. We're just going to label this. So we're going to label each molecule. The 6CO2 is the carbon dioxide. Okay. The 12H2O is the water. And then we've got glucose. I mean, you can just type in sugar too if you want. Glucose, specifically glucose. Oxygen. That's oxygen gas that's created as a byproduct. And then finally, water on the other side. Yeah. All right, so does photosynthesis look like cell respiration going backwards? You bet it does. Yes, it does. All right, so the chemical process takes place in 2 steps. So even though it's shown in one reaction above, there's the light reactions, and then there's the dark reactions, and we're going to talk about those next. All right, so in the diagram above, this kind of shows what's going on inside the chloroplast. We can see that the light reactions are taking place over here, and these would be your dark reactions taking place in the Calvin cycle. So I know there's a lot going on here. Let me just break it down real quick for you. The light reactions, notice the light is coming in. I'm going to draw on this. Let's see. Oh, no, I'm not, because I need to label it. So, let's just do this. Let's label the diagram, and then we can talk about it as we go. Okay, so color carbon dioxide red. And so carbon dioxide is what we breathe out. That's the byproduct of our metabolism. And it's going to enter the Calvin cycle. And that's where it gets reduced and turned into a sugar. So remember, reduction is where you gain electrons. And so carbon dioxide is highly oxidized. We need to reduce it, give it some free energy, give it some electrons. And those electrons are going to come from the light reactions. Remember, the light reactions are how sunlight excites electrons, and then those electrons are transferred to carbon dioxide, reducing it, turning it into sugar. Ooh, I know it's a mouthful. All right, so we're going to take water, and so our water is going to be the water is the, what would you say? It's the reservoir of electrons to get excited. So the electrons that actually get stimulated by the light energy come from water. We strip them away from water and then we charge them up in the light reactions. Okay, we're going to take the light energy and color it yellow, orange, whatever. So here's our light energy is coming into the light reactions. That's where all the magic is happening. And then we've got sugar is green. So sugar is the byproduct. So remember, the light reactions come in. They're going to charge up the electrons from water. Those charged up electrons get carried in these two cellular batteries that get charged up. They bring them over to the Calvin cycle and drop their electrons off on carbon dioxide, which eventually gets reduced into sugar. And then finally, what happens, I don't have orange necessarily, so I'm going to use red. Don't worry, guys, I'm not going to mark you wrong if you use red instead of orange. So carbon dioxide I know is red and light was yellow. I don't have orange, but we'll do the best we can. So we'll take oxygen and color that yellow. Maybe I'll yellow and maybe I can make it orange. Haha, there we go. A little yellow mixed with red. And so oxygen is the byproduct of splitting water and stealing its electrons. So when we strip away the electrons from water, we get oxygen gas as a byproduct. We also get left with a couple of protons. That's where those H's go. And those protons, we talked about those in our photosynthesis lecture. We won't go into detail on that right now. But the oxygen is produced when we strip the electrons away from water, charge them up, take those electrons, transfer them to a cellular battery, charging the cellular battery up. The cellular battery then drops off the electrons into the dark reactions. Those charged up electrons reduce the carbon dioxide into sugar, a more energetic molecule. And so really you just have to follow the energy in these sorts of systems. So the light reactions, those happen in the thylakoid. And the Calvin cycle occurs in the stroma. So remember how I said the cytoplasm-like substance that is inside of the chloroplast? That's the stroma. So you've got the light reactions take place in the thylakoids. Calvin cycle takes place in the stroma. All right, so we're going to do 2 experiments in this lab. We won't be able to do them from home. One of them is chromatography. The other is a light gradient experiment. And so what we're going to do is we're going to use data from a previous lab that was done in person, and then we're going to plot it out on a graph. So basically how this works is that we take Alodia, which is this water-based plant, and we place it into a little tube with water in it, and we cap it, and then we put these tubes, so we make a whole bunch of these tubes with these plants in them, and then we place tubes at different intervals from a light source, and I'll show you, there's a diagram right here. And so here's our light source, here's our tube with, I don't know if you guys can even see my marker or not. Here's our tube with Alodia. Here's our light, so here's my light source. And then here's my tube with my Alodia in it. And so what we've done is we take these tubes and we place them at different intervals away from the light source. And so you have tubes that are really close to the light source that are going to be receiving tons of free energy from the light source. And then we're going to measure how does the intensity of the light affect the rate of photosynthesis. And so what this begs, well, the question it begs is this: okay, so what's our independent variable and what's our dependent variable in this experiment? Well, in this experiment, we can measure the amount of dissolved oxygen produced. So come back up here to the top. When Alodia starts to photosynthesize, oxygen gas is just a byproduct. It's a waste product for plants. They don't need it. It's what's left over after it's taken the electrons away from water. And so Alodia will produce this oxygen gas as a byproduct of photosynthesis. So we can measure how much oxygen is in the water at given intervals to see how fast this plant is photosynthesizing. And so what do you think the bubbles are that are being produced on this plant? Right. Okay. So what we do is we're gonna take and put these, basically if you're reading this, so Elodia is placed in test tubes at different distances from the light source. The different distances from the light source represent the independent variable, because that's what I can manipulate in this system. I can say, okay, how does intensity affect the rate of oxygen production in Elodia? And so the light intensity is my independent variable, because I can actually manipulate that. I can put the plants at different distances, and that changes the intensity at which they're receiving the light. The dependent variable, though, is how much oxygen is produced, because I can't actually physically change that. I can change the independent variable and I can measure its effect on the dissolved oxygen produced, but I can't physically like make a plant produce more oxygen. It does that on its own. And so the measured output of this experiment is the amount of dissolved oxygen and the change in the dissolved oxygen over time. Now, remember, I think, where is it? There's a control Elodia tube wrapped in aluminum foil. And so think about that. If we take a tube, put the plant inside of it, wrap it in aluminum foil and stick it in front of the light source, is it getting light? No. So it shouldn't be producing oxygen. So does a plant have mitochondria? Yes. Do mitochondria use oxygen? Yes. So what do you think is going to happen to the dissolved oxygen in the tubes that are wrapped? Let's just think about that. All right, so below is the setup for the experiment. We used it's PAR, so it's the number of how we determined the light intensity, right? Because that would be our independent variable. Is we used a PAR meter, a photosynthetically active radiation meter. And so what it measures, it doesn't measure wavelengths, it measures particles striking a given area over a given interval of time. So how many light particles are striking this much area over a given interval of time? So that's what this is. How many light particles over a surface area over an interval of time? I already said that. Okay. So we use the light meter to measure this. And so what we did was we set it at 400 particles per little square meter per second. And that was really close to the light source. And then we backed one up a little bit. That was 200, 100, 50, 25, and then one that was wrapped in foil that was 0. Okay. And so that's the experimental setup there. And then in the table below, what we did was we are reporting the results that were obtained for this experiment. So lucky for you guys, you don't actually have to sit for 30 minutes and wait for this thing to take place. It's already been done for you. And there's a cool little photo of this guy that's like measuring dissolved oxygen in the Salt River, which is something you can do. All right, because dissolved oxygen, obviously fish need it to survive. And so it's a good question is, how much oxygen is available for the aquatic life in our rivers? Okay, so here we have it. What you'll notice is up here, they set the exposure at 400, 200, 150, 25, and 0. In our table, though, it was listed at 350, 160, 80, 40, 20, and 0. So pretty close to the same, but don't get confused by that. That might as well say 400, 200, 150, 25. Okay, those are just different intensities of light. All right, so what we have here is column 2. This is a little bit confusing. Column 3 represents the dissolved oxygen in each tube when we started. That was right when we started. The column 2 is how much dissolved oxygen is present when we finish. So after like 30 minutes of sitting in front of this light source. And so what column 4 is it's, okay, what is our change? What is our delta of dissolved oxygen from our start time to our finish time? And so when we're calculating the change, we start with what we end with, and we subtract that from what we started with. And so we finished with 13.9. So I'm going to write this in. You guys don't have to. 13.9 minus 7.0 is equal to, let's see, I'm sure it's 6.9. So that's our, and it's plus 6.9, right? And so I'm just going to put that right there. So I took what I finished with, subtracted it from what I started with, and I ended up with 6.9. Okay. the next one. So remember that we had 2 tubes for each interval of light, 2 tubes at each interval. So 2 tubes being exposed to 350, 2 tubes being exposed to 160, 2 tubes for what. So let's see what the other tubes change was. And I can pretty much just do this in my head for a lot of these. And I'm gonna tell you guys to pause the video and you guys run through all the calculations. I'll do it too, but I don't want to like waste your time and do them all for you. I want you guys to learn something too with some hands-on activity. So let's go 12.6 minus 7.0 is equal to plus 6.6. Okay, that's my change in dissolved oxygen content for 350. Now I'm going to pause this video and I want you guys to do the same thing. Go ahead and fill out the table, finish the calculations, and we'll pick right up and then we'll start with our graphs after that. Okay, so I filled in the table. Try not to copy me. I would appreciate it if you did your own work. And so we've calculated the change for different intensities. So we've basically calculated our dependent variables here. And now we're going to plot those out on a graph. And so what I'm going to do is I'm actually going to do this in Excel. I'm going to show you guys how to do this in Excel. And then so we're going to plot the x-axis. Now you guys can do this on this graph here. You can write in your little axis labels. What you're going to want to do is you're going to want to put from 0 all the way here. I'll even help you. I'll help you guys out here. So if you're going to do it on here, you can take this from 0, and then I think we had 20, and then I think we had like 60, and you can put your axis labels here. What I want to do though is I want to give myself enough space, and so I think I'll go 0, 20 on this second one, and then 60. I'll make sure I'm doing the right intervals. 20, 40, 80. All right, 20, 40, that should be 40. 20, 40. Skip 2, 80. Skip 2, 160. And then let's see. And 20, 40, 60, 80, 160. And then, why am I drawing a blank here? 350. And then 350. Yeah, so that's my x-axis right there. Boom. I have them spaced out pretty good. And then y-axis is going to be numbers. So that's going to be the change in dissolved oxygen content. Now, here, let me give you guys a hint here. So set your 0 to be like right here. Because if you remember, one of our measurements was negative, and so we got to be able to represent a negative change in dissolved oxygen. Now what we can do to figure out what our what our spread should be on our what would you call these, like our axis points or y-axis point. Spread's it's gonna go all the way up to, it looks like 8, and then all the way down to negative 0.2. And so what I can do is say, okay, maybe like 2, 4, 6, 8, and then I'll go negative 2 down here. And so you guys can do that. So you can go 2, 4, 6, 8. So I'm gonna put 8 up here. 2, 4, 6, 8 right there. And then I'm gonna put a negative 2 down here. Right there. Cool. And then you guys can fill in the rest. And so you can use this to plot if you want to, you can use this and just fill this out, do your scatter plot and stuff on the PDF editor. But for those of you that are trying to figure out Excel, let's go ahead and do it in Excel. So I'm just going to pause it. I'm going to open up my Excel. What I'm going to show you is I'm going to copy this data over, and I'm going to correlate it in a column next to the these x-axis variables. And I'll show you here in a second. So I'm going to pause the video. And then when I come back online, I will have copied this over into Excel. All right, so let me just change what I'm sharing. So I'm gonna come down here. Oh, I just stopped my video. Okay, let me share my Excel spreadsheet with you. Okay, so all I've done is I just copied over the one column that has my independent variable, my different intensities of light. And then in the column next to it, I just wrote down the change in dissolved oxygen. Now I don't even have to click on this guy when I go to make my graph. So check this out. We're going to make a graph. So we're going to select all this, just make a big box around it. And then we're going to go up to insert, and we're going to come over to our recommended charts over here. And so you'll see this one right here, insert scatter plot, and that's what we're going to do. We're going to go scatter plot. Now you can do all these weird looking ones, which are kind of funky. They look gross. Let's just do this one. And so this one just shows, it plots both measurements at each interval. And so what you're getting is you're getting both measurements taken at the 0, both measurements taken at the 20, both measurements taken at the 40, all the way up to the top here. Now, what we can do now that we have this chart is we can add in our information. We can add in like our chart elements. So what we can do is we can add in our axis titles. So let's go primary horizontal. So what are we looking at here? We're looking at light intensity using our par meter. So that's our light intensity. And now we need to add another chart element. So we'll come up here and we'll do axis title primary vertical. So what are we looking at over here? We're looking at our change in dissolved oxygen content. It's our change in dissolved oxygen content. And then chart title, this is, we say, the effect of light intensity on rate of photosynthesis. Boom. Effect of light intensity on rate of photosynthesis. We have our light intensity plotted here. We've got our change in dissolved oxygen content on the y-axis. The only thing we're missing is actually a trend line. And I know that it asks you to do this to kind of like draw a trend line on the graph. And we can actually have Excel do that for us. It's pretty robust. So we're going to come up, click on our graph. So we have it selected. Then we're going to come up to, chart design. And we're going to add a chart element. So just like we did our titles, we're going to come down here to trend line. Now what I want to do is no trend line, linear. Linear doesn't really work because it's not shaped like a line, it's shaped like a curve. We got linear forecast, moving average. None of those look good. So let's go more trend line options. And we'll come over here. What happened to my trend line options? I want standby. Trend line. There we go. Okay. I don't know why it didn't show me this the first time. So trend line options, we're going to go polynomial and go third order because that's going to snap it closer. If we go higher orders, this is going to look all funky. So go like third order. Makes it look more logarithmic. And that's our trend line. So what we notice is that we kind of cap out, like we can only do so much photosynthesis up to a certain light intensity, and then any light intensity after that is just wasted light energy. It's not going to be able to be absorbed by our photosystems. All right, so this is our graph. I'm going to switch this back to our other screen now. So let me make sure that I'm actually sharing it. I'm sharing that. Now I gotta switch this back. Stop sharing. Then I'll share my Chrome. Okay. So let's see. We did our graph. If you guys did it here, that's fine too. It says do a scatter plot. And then it says sketch a best fit, which we did. And then take a picture of our graph and submit it along with this file. What we can do is we can actually just like copy and paste it into the bottom. You guys can copy and paste it onto this blank page here and then save it all as one file. And then it's only one upload for the whole thing. And that's what I would do. Okay, so that graph is done. Moving on to our next. So paper chromatography. We know that plants absorb light. Anything absorbs light that has color. So like, you know, my black shirt, if I walk out in the sunlight, is going to absorb every wavelength of visible light and reflect none back. And so my shirt will warm up because it's collecting that light energy. But it's because it has pigments in it. And so these photosynthesis pigments collect the light in our photosystems. And so our photosystems have their own built-in pigments. Some of them are like chlorophyll and stuff. And so what I'd like you to do is go on this YouTube video, so click it and follow the link and go watch this video, and it shows you exactly how chromatography is done. I'm going to talk through it briefly, but I'd like you to watch the video too. All right, so first you have to take the chromatogram, which is the piece of paper like you see down here. And what you're going to do is you're going to take in and you're going to concentrate down at one end of the chromatogram extract from your leaf. It'll be kind of a liquid, probably suspended in ethanol, something that evaporates quickly. And so it'll be like this green liquid that's an extract extracted from the leaf pigments. So you take it and you're going to concentrate that solution into one spot right at the end of the chromatography paper. And then what happens is you take it and you dangle this chromatography paper into a beaker that has solvent in it. And the solvent will dissolve just like a paper towel. The solvent will wick up the chromatography paper. And as it passes by our pigments that we dried onto the paper, it will dissolve them. And then just like a paper towel, like water runs up a paper towel, the solvent will move up the chromatography paper. And what it does is it spreads out the pigments based on their density and their weight. And so if it's a very light molecule, something that's not very dense, it's going to go really far on the paper. It's not going to have as much friction, and so it's going to come further with the ethanol or with the solvent. The heavier pigments are the ones that get left behind because they're like dragging, they're too heavy. And so what we're going to do is we're just going to label the pigments on the chromatography based on the YouTube video up here. We can use the timestamp 303 to find exactly where those are. Now, I'm not going to do it. You guys are going to do that. And it's not that hard. And so you'll just label chlorophyll A, B, xanthophylls, and carotenes. So this next question, I'm going to help you guys answer it. And I think you probably already know the answer. Is chlorophyll the only pigment in the spinach leaves? Well, no, we've got the xanthophylls and the carotenes. So no, it's not the only one. Why do you think the leaf needs additional pigments? I'll tell you why. Maybe you guys can come up with the answer, pause it, and think about it. Why does it need extra pigments? Why not just chlorophyll A and B? To capture the extra wavelengths of light that are captured by chlorophyll A and B. So chlorophyll A and B are only going to capture certain pigments or certain frequencies of light, and they'll reflect other ones. So these other pigments are going to do a good job of capturing the additional wavelengths of visible light. It's basically a way of it's sort of the difference between wearing like a white t-shirt versus wearing like a red t-shirt. If you wear a white t-shirt, it's going to reflect every wavelength of light. You wear a red t-shirt, you're going to absorb the red wavelengths, or you're going to absorb every wavelength except red. And so it's the same idea here, is that the plant is spreading out what sort of wavelengths it can absorb by using different pigments. And so the more wavelengths you can absorb, the more energy you can absorb, and the more energy you can process. All right, and so moving on. So now we have our final questions in this lab. I'm not going to actually fill in these blanks for you, but I will talk through the questions and you can fill them out. So how does measuring photons of light different from measuring light as a wave? Well, light can be measured as a particle or a wave, and that's the difference. When you're using the photons of light, you're measuring it as a particle. When you're measuring it as a wavelength, you're measuring it as a wave. Two different things. Give the light level at which compensation point was reached, and then explain what compensation point represents. Let's talk about that for a second. So the compensation point is the point at which, and I'm not sharing, I'm see if I can switch my share to, I don't know, I'm not a huge fan of this WebEx. But anyways, maybe what I'll do, let me just do this real quick. I'm gonna pause it. Okay, so I'm back. So let's make sure, yes, I'm sharing the screen. Okay, so we're answering this question. Give the level of light which compensation is reached. And so what it's asking you what the independent variables measurement would be, where the amount of oxygen being used up by cell respiration by the mitochondria equals that's being produced by the chloroplast in photosynthesis. And the way we find that is we come down to our graph and we look and see where the line crosses the 0 at. And so what we can see is, okay, we got 50 here, so that's 25, so probably, and then there's our 20, which is a little bit above. So I'm going to say like 15. So 15 is going to be what I would say. So we come up here. The compensation point was reached at 15 micromoles of photons per meter squared per second. Okay, so 15 there. And then explain what compensation point represents. Well, it's when the amount of oxygen being used by the mitochondria matches that is being produced by the light reactions in photosynthesis. So it's where you're breaking even, basically. All right, give the light level at which the saturation point was reached. So let's come back down to our graph and figure that out. The saturation point is where you can no longer create more light energy from, or you can no longer photosynthesize any faster. It's basically a good way of looking at saturation. It's like, if you've ever heard of like a governor on a car, like some cars can go faster than 120, but you know, there's a chip inside the engine that will not let the car go past 120 miles an hour, no matter how much gas you're giving it. This is the same thing. There's a point on this graph where you're no longer producing dissolved oxygen, no matter how much more light intensity you add. And so that saturation point is somewhere up here. We can see where it kind of like tops out. And so I'm going to say it's probably, we can see it's a very peak here, probably about 200 nanomoles or micromoles per meter squared per second. So that's a saturation point. It was about 200 micromoles per meter squared per second. What does it represent? I already told you. It's the rate. It's the point. It's the limit of photosynthesis. So it's the upper limit. It's the ceiling of photosynthesis. It's the most. Yeah, it's the highest rate that photosynthesis can take place. Any higher intensity of light, you can't make any more oxygen. So explain why the change in oxygen was negative when the elodie was placed in the dark. Well, that would be cells have mitochondria, and so the mitochondria are using up oxygen, right? You're still the plant cell still needs ATP. Its mitochondria is still going to be active even when it's dark out, even when there's no light. The mitochondria are still working, burning up oxygen, making ATP, and so that's going to drop the oxygen levels in the water. And so what's happening is cell respiration is using up oxygen, and photosynthesis is not producing any oxygen, so you get a negative change in oxygen when it was wrapped in the tinfoil. Why do some molecules travel faster on chromatography paper? Well, it's because they're smaller and they weigh less. They have less drag. And then what is a pigment? A pigment is any molecule that absorbs a wavelength of light, period. And guess what? You guys are done. And notice how I put my graph down here. So all I did was, I'm a rookie. I'm okay with computers. But all I did was I did it in Paint. Like I took a screenshot of Excel, opened it up in Paint, cropped it, saved it as a JPEG. And then I was able to take up here with this right here, this button. And I was able to take my JPEG that I saved and just put it right into my PDF. Now check this out. Since we're all done, it's really cool. We can just go download, watch this, download. So it downloads it as a PDF, and then I can open it up, and then look, boom, it's got all my work. Hey, how cool is that? And then you can just save it and submit this. So hopefully this video was helpful to you guys. I know a lot of you are using them. Some of you guys aren't, and that's totally fine too. All right, had a lot of fun. I'll see you guys next time.